Hello lovelies, in this paper we're going to be going through your Combined Science Foundation Biology paper 2, the paper that we've written for 2024.
The diagram shows the evolutionary tree of part of the big cat family, Felidae. Wants us to state which big cat is most closely related to the jaguar. On the family trees like this, the most closely related ancestor will share a branch or be on the closest branch to the one you're being asked about. So in this case, it shares a branch with the lion, so you can see they've got a join there, so that's our answer. The image below shows a picture of a clouded leopard. Now it wants us to state the genus of the clouded leopard. The genus is always the first part of the binomial name because we use the genus and species, taxonomy groups to make the binomial name. So Neophelis is the first part of the binomial name for the clouded leopard. And then lastly, state the species name of the snow leopard. So the species name is the binomial name. So we need both parts of the name. We don't just say the second part. It doesn't make any sense without the genus. That's why we have the binomial name. It has to come in two bits. So if we're doing the snow leopard's binomial name or species name, it's Panthera uncea. Common mistakes made with these questions is being careful when copying. You are copying down Latin names, so be careful with spelling, but also make sure you use capital letters and small letters where you should as well. And remembering that the species name is the binomial name, so you need both parts. The snow leopard is quite different from the other cats. It lives in very cold, snowy, mountainous habitats. Suggest a feature of the snow leopard that helps it to survive in its environment. So this is an adaptations question. That's what it's really asking here, about features that help an organism to survive. So it's also a suggest question. So we need to use our knowledge about adaptations and also the question information which tells us it's very cold, snowy, mountain habitat. So we need to give specific adaptations to this information about it being very cold. So adaptations we'd expect to know or have learned for cold environments would be something like a fat layer to insulate, thick layer of fur or hair, or having a small surface area to volume ratio to try and reduce heat loss. So I'm going to go with a thick coat of fur to reduce heat loss. We need to make sure we add how it helps it survive. So it's only one mark, but you can't, I'd rather than just put a thick coat of fur, making sure you've added that extra bit so you're linking it directly to why it's an adaptation to the cold and how it helps it to survive. Unlike all other cat species in the same genus, the snow leopard cannot roar. There is a debate about whether it should have its classification changed. So this would mean it would not be in the same genus anymore as the others. Describe a method scientists could use to determine if the snow leopard is significantly different enough from the other cats to be put in a different genus. So when it asks to describe a method, it's talking about what are the steps in the process and how would we be able to tell or decide if something is significantly different in terms of relatedness from other organisms. So normally we compare and talk about similarities in their DNA. That's how we make these phylogenetic trees. And how do we look for differences or similarities? We compare the base sequences. So we need to sequence the DNA of the cats in the genus, so all the cat species in the genus, and the snow leopard as well. And then we would need to compare the base sequence of their DNA to see how different they were. Variation may arise due to the geographic isolation of a species. Geographic isolation of members of one species can lead to a new species evolving. Name this process. So this one is just asking us, what is it called when a new species is formed? So the word evolving is already in the question, so it can't be evolution. The answer is speciation, and the clue is in because it has the word species almost in the name. But evolution and natural selection is the process by which evolution happens. And evolution doesn't always necessarily mean that a new species will evolve. It just means a change in a species over time. But if it happens to two different populations and they change so much that they can no longer interbreed with each other, then we get the species, because our species definition is a group of similar organisms that can breed to produce fertile offspring. So if they can't breed to produce fertile offspring anymore, then they will be a different species. So only then do we get speciation, and it normally happens when the populations are separated, like what it's talking about with geographic isolation at the start of the question. So they have to be separate for a time, let natural selection and evolution happen over time in separate populations that can't breed together, and then eventually they'll be changed so much over time that even if they're put back together, they wouldn't be able to breed to produce fertile offspring anymore, and so we've created this new species. Explain how geographic isolation of members of one species can lead to a new species evolving. So now it's asking us to explain, like what I was just saying, how geographic isolation, so how the separation of those members of one species and then natural selection and evolution can then lead to that new species evolving. So we need to link this idea of isolation and then how a new species forms from it. So the process is speciation, and we've got this idea of like there's two populations of a species, A and B. They're one species, but they got separated geographically for some reason. And so either side of this, in these two new environments, we're going to have different environmental conditions. And then natural selection is going to take place in either side of the new environments. And if we're forming a new species, then it means they must not be able to breed with each other anymore. So that is enough really for 3 marks. This isn't a really long answer question. We're not having to go through the whole process of like evolution. We're just giving an overview for 3 marks. Then we're going to say that the different areas are going to have different environmental conditions. We're going to briefly describe what natural selection occurs so that only the best adapted members in each environment survive and breed. And then what makes them new species is eventually the two populations can no longer interbreed, so they are new species now. Now, I've probably said more than enough for 3 marks, but this is still a decent level of detail and kind of what you need to be talking about if you're talking about speciation. Beta thalassemia is a recessive inherited disorder which reduces the production of hemoglobin in the body. The family pedigree shows the inheritance of beta thalassemia in one family. So my first thought is, what is a recessive inherited disorder? It's the one that needs 2 alleles to be the same to be present in the phenotype. So it's the one where you have to have 2 lowercase letters, for example, in order to have the disease. And if we're thinking about maybe a question we might get asked later, we might not, but I'm thinking, okay, if it reduces the production of hemoglobin, that means there'd be less oxygen being transported if we don't have as much hemoglobin in the body. So we've got the parents in the first generation, then we've got their children and their grandchildren in the third generation. Tick the row that correctly identifies the sex and genotype of person B. So we need to go and find person B on the family tree. So they are a filled-in circle, which means that means from the key they are a female with beta thalassemia. So it's just looking at sex determination here. So the sex chromosomes, we're not talking about the alleles for beta thalassemia at this point. So we know they've got to be female. And so it's got to be one of those 2 rows. But we've got either XX or XY. So this is really just a question about, do you know the sex chromosomes for male and female? So XX is the sex chromosomes female and XY is male. So we're ticking that bottom row. Explain why sperm determine the sex of offspring at fertilization. So the reason why is what we need to try and say here. Why does sperm chromosome determine if an embryo has XX or XY? And this is simply because sperm can either contain an X or a Y, depending on which chromosome is present out of the 2 options. Eggs can only contain X chromosomes because female cells can only donate an X chromosome to each gamete. So if you have a Y chromosome present in the sperm, then the fertilization will happen and you'll get XY and therefore a male offspring. If you have an X in the sperm, then you'll get XX and you'll have a female offspring. So this is basically what we need to say. We need to say that sperm can carry a Y or an X, and having a Y chromosome determines male, and if you don't, then you're female. The gene responsible for beta thalassemia can be represented by the letter B, capital B. Capital B represents the dominant allele, and little b represents the recessive allele. Both individuals F and G in generation 2 are heterozygous for beta thalassemia, so they have big B, little b. Use the diagram and your knowledge to explain why. So we need to be able to explain why they must both be big B, little b, or heterozygous, and what evidence can we get from the family tree. So if we find F and G, we can see that they do not have the disease, but they have had an affected child. So this is very common with recessive disorders. In questions like this, they'll get you to go and find two parents that do not have the disease and they've had a child that does have the disease. So that must mean they both have to be heterozygous because they both must have a copy of the lower case allele, the recessive allele, in order to give both of them to give a copy to child I in order for child I to be little b, or double or homozygous recessive. So we can always start our explanation by saying they are unaffected, but they produced a child, in this case child I, who is affected. So we can say they must both have had at least one recessive allele in order to both pass on the recessive allele to child I, but obviously they are both unaffected, so they must also have had a dominant copy. Use the Punnett square to calculate the probability that the next child, individuals F and G, could have beta thalassemia. Use capital B to represent the dominant allele and use lowercase b to represent the recessive allele. And they filled in the parental genotypes for us. And then we fill it in as normal, so 2 capital B's, 2 squares that have large b and lowercase b, and then 1 square with 2 lowercase b's. So that then needs to be circled to show that we understand that that genotype would produce an affected child, and then we need to give the probability. So it's 1 out of 4 or 25% chance that the next child they have will have the homozygous recessive and therefore have the disease. So as you can see in the mark scheme, just doing the Punnett square without circling the little b and giving the information doesn't get you the mark. So make sure you add this every single time you do a Punnett square question. You need to make sure you circle the one thing that the question was asking you to look for the probability for and then label it with the probability. Explain why it's important for people like C or E to have a pedigree analysis in case they decide to have children in the future. So we can see C and E are children of A and B. So their mum has beta thalassemia and so does their brother. It says use percentages or ratios to help illustrate this. So we're going to need to sort of explain what the percentage chance might be of them passing it on depending on what the analysis shows. So why is the reason why they like to have a pedigree analysis? And it's because obviously they might be able to pass on the recessive allele. So they're going to want to know what the probability is of their offspring having the beta thalassemia allele. So if we think about the percentage chance, obviously we've just seen that it could be 25% chance of having a child with beta thalassemia. There could be 50% chance of passing on a recessive allele if you are heterozygous. And if they're homozygous dominant, so they have 2 capital B alleles, 2 dominant alleles, then there's 0% chance of them passing on the affected allele to their offspring. So it's all going to depend on what alleles they have and then what alleles their partner has in order to determine what the probability is of having a child with the beta thalassemia allele or having the disease. But only with analysis will they be able to show that, so then they can obviously make those decisions. So an analysis will determine the probability of passing on the recessive beta thalassemia allele. It could be 0% if they're homozygous dominant, or up to 50% if they are heterozygous. A father with the genotype capital B, so homozygous dominant, and a mother with the genotype b, or homozygous recessive, plan to have several children. Explain why none of their children will have beta thalassemia. So explain why none of their children, the reason why none of their children will be little b. And when it comes to these questions, it's often just easier to draw a Punnett square rather than trying to write it out in words. And often you will get marks for that as long as you annotate it correctly. So let's put those into a quick Punnett square. So we've got 2 capital B's and 2 little b's. So it shows that 100% of their offspring are going to be heterozygous. So they're going to all have at least one dominant allele and one recessive allele. So they'll all be carriers, but none of them will be homozygous recessive. So none of them will have the disease. So you can say none of them will have 2 recessive alleles. You could say they can only get 1 recessive allele from mum, and so therefore they can't have 2 because the father can only donate capital or dominant alleles. However you want to explain it, you've got some options in the mark scheme, but if you want to do the Punnett square, that's fine, but you would need to have this extra little bit of text that I've written next to it just to make it clear that you understand that 100% of the offspring are heterozygous, or something like no child will have 2 recessive alleles to have the disease. Students were asked to estimate the number of buttercups in a sports field that is 50 metres wide and 90 metres in length. They used a 0.5 metre by 0.5 metre quadrat like the one shown in the image below and placed it in an area of the field that contained some buttercups. 1 buttercup flower has been circled for you. Identify the number of buttercups that the student should record in this quadrat. Tick 1 box only. Okay, so we've got the circled one, and then we've just got to count the rest of the number of buttercups we can see inside the quadrat, and then we've got to pick which one of those numbers is right. So we've got our 1, the one that's circled, then 2, 3, 4, 5, 6, 7, and then 8 is there down the bottom. So the best and sort of easiest way to do this, and what you should have done this when you've done this practical before, is kind of go along each row of the quadrat, and then you don't miss any. Make sure you go along each row. Make sure you can count every single time you see a buttercup. 3 I've crossed out, don't count because they're outside of the quadrat. So our answer then is 8. Use the quadrat to calculate an estimate for the total number of buttercups in the field. Okay, so to do that, first of all, we need to calculate the area of the field. So the area of a rectangle is obviously just length times width, so 50 times 90. So that gives us 4,500 metres squared. Then we need the area of the quadrat. So that's 0.5 by 0.5 metres, so that gives us 0.25 metres squared. So to do the final sum, we need the 8, which is the number of buttercups in this quadrat, divided by the area, so number of buttercups per this area that we counted them in, which was 0.25 metres squared. And then we're going to times that by the whole area of the whole field. So that gives us 144,000 as an estimate of the number of buttercups in total on the field. Describe a more accurate method the student should use to estimate the number of buttercups in the field. So the clue here comes from the part of the question where it said they placed the quadrat in an area of the field that contained some buttercups. So they found some buttercups and then put the quadrat down. So that's biased, and we know that that's not the way that you should do this practical because it needs to be random. So it wants us to describe, so give steps in a process, give the detail, a more accurate method, so one that would be random, for example, and way they should use to estimate the number of buttercups in the field. As well as being random, the placement of the quadrat, they only did 1. So us using that 1 quadrat was not acceptable. You have to repeat it, do it quite a few times to get a representative sample of the area, and then you would calculate the mean number of buttercups in all of those different spots that you put down in the field, and then you do the same calculation we've just done. So for 3 marks, we're going to have to describe how we're going to do it randomly, how we're going to do many of them, and then that we're going to calculate a mean from that. So my first mark is going to come from saying use a random number generator to produce random coordinates in the field. Now, there is just a mark in the mark scheme here for it being or mentioning that you should do it randomly. So however you've said the word random, as long as you've got it in there and you've made it clear that you would do something in your method to make your placement of the quadrat random. You'd place at least 10 quadrats. So this is about there being a large number. So that's the mark in the mark scheme. And then we would calculate a mean in order to then across those quadrats, and then we would estimate our number. So always in these practicals, when they ask about improvements, how can it be made better, more accurate, you need to be thinking about large number of quadrats. Did they do that? Yes or no? Did they do it randomly? Yes or no? And you can always say repeat and calculate a mean, okay? Because that will always get you a mark here, because the more you do that in any experiment, that's going to make your results more reliable, okay? So when we're thinking about this practical in particular, these are the questions and these are the things they always want you to say. Buttercups have a shiny surface to their petals which reflects UV rays into the centre of the flower. This increases the temperature of the flower. It has been shown that bees prefer to visit warmer flowers as it helps them to maintain their body temperature. Explain how the shiny surface of buttercups is an adaptation. So remember, an adaptation is a feature of an organism that helps to increase its survival or reproduction, because then it's passing on its genes to the next generation. It wants reasons why that shiny surface is going to be an adaptation. And obviously it gives us information in the part of the question that says that the bees prefer to visit warmer flowers. And it's the shiny surface that helps to increase the temperature of the flower. So if your flower is warmer, more bees are going to come and visit, which means you get more pollinators. And if you get more pollinators coming to the flower, then that increases your chance of sexual reproduction. It increases the pollination that's going to happen between carrying your pollen to other flowers as well. So the first mark's going to come from saying that we understand that it increases the temperature, which attracts more bees. Don't get any marks just for talking about that it increases the temperature, because it tells you that in the question. So you've got to link the increase in temperature to more bees coming and visiting the flower. And then our second mark is coming from how we're saying this is an adaptation. So remember, how is it helping it to survive or reproduce? So pollination by bees increases chance of successful sexual reproduction. Even if you just said sexual reproduction, or it increases the chance of pollination, that's fine. The students noticed that there seemed to be fewer buttercups around the edges of the school field where there were lots of trees and hedges. Suggest a reason for this. So this is a suggest question. They're often the hardest ones on the paper. You're going to need to bring in your own ideas and information given to you in the question and put it together to provide an answer. It's not necessarily going to be something, just a piece of content or knowledge that you can just regurgitate. So it said there's lots of trees and hedges around the edges of the school field, and there were fewer buttercups there. So we need to link this idea of why having lots of trees and hedges might reduce the number of buttercups near them. So there's a couple of options here, and this is quite common with suggest questions. There's not normally only one right answer. So we could mention that the idea that these trees and hedges are going to shade that area of the grass so there's less light for photosynthesis. Or it could be due to competition, so bigger, larger plants nearby, there'll be less nutrients available and less water available in the soil, and therefore the growth of the buttercups would be worse. So because this is an explain, so we need a reason, we need to make sure we do say not just that it's shaded. For 2 marks, we need to say shaded or less light, and that means less photosynthesis, so they're not going to grow as well. Or the competition, we say obviously less nutrients or water, and therefore they won't grow as well because of that. So I picked the light answer, so the trees and hedges create shade, so there is less light available for photosynthesis. The diagram shows a neuron. Name one structure found inside the cell that would not be found in a prokaryotic cell. So hopefully the word prokaryotic makes us think of bacteria, and the fact that a prokaryotic cell means it has no nucleus. And just to check, it says one structure found inside the cell that's not found in a prokaryotic cell. So what would be in this one, this animal cell, but not in a bacterial cell? And that's hopefully an easy one, that's just the nucleus. You can also have the mitochondria, which you can see in the mark scheme, and that's because obviously mitochondria are too big, they wouldn't be found inside bacterial cells because they're about the same size as bacterial cells. Also, just to think that I highlighted inside the cell, so we're not talking about cell walls or cell membranes here because that's talking about what's inside the cell, but also an animal cell has a cell membrane, and so does a bacteria, and a bacteria has a cell wall, but it's asking what would not be in a prokaryotic cell, so that wouldn't count either. Name the type of neuron that sends signals from receptors in the eye to the brain. So when it says type of neuron, it means either the sensory, the relay, or the motor neuron. Those are the three types of neurons we've learned about. And it's asking which one's going to be taking electrical signals from the receptors in the eye to the brain. So that's the direction and where it's going from and to. So if we think about the different types of neuron and what they do, so motor neurons are always attached to effectors or muscles, think movement. Relay neurons are found inside the spinal cord in the central nervous system, and the sensory neurons are the ones that are attached to the sense organs. So the eye is a sense organ, and also receptors are always found in sense organs, so it must be the sensory neuron that's going from receptors to the brain. Describe how impulses travel between neurons. So describe hows are always like steps in a process, steps in order. How does this happen? Tell me how it happens. And if we're going between neurons, that's from one neuron to another. So this question is all about synapses. It's being able to say, how does it cross the synapse, basically? How does an electrical impulse travel from one neuron to another across that synapse gap? And so it travels by a chemical diffusing. So that chemical is called neurotransmitter. If you can't remember that, just talking about the fact that it's a chemical is enough. And this idea that it's released from one neuron, and as a chemical it diffuses across the gap, it will bind to the next one and then cause that electrical impulse to then carry on in the next neuron. So there's only 2 marks here. This could be a 3, even 4 mark question if you get the detail correctly. So the first mark comes from saying that the chemical neurotransmitter, you don't have to include that, is released. That's the first point. And then the second mark is coming from saying that it's diffusing across the gap or across the synapse. We can get a mark for saying synapse here because it didn't say that in the question. So that's where we're going to get one of our marks from. This idea it's got to have you've got to talk about diffusion and the fact that it diffuses across the gap. That's how it actually moves from one neuron to another. The speed of impulses being passed between neurons is important as it determines reaction time. The average gap between 2 neurons is 32 nanometers, and the average time it takes for the impulse to pass between 2 neurons is 1.6 times 10 to the minus 7 seconds. Calculate the speed of the impulse crossing the gap between the 2 neurons. Use the formula speed equals distance divided by time, and give your answer in meters per second. Now, this equation may be familiar to you because it's obviously from the physics specification, but they do this sometimes across the sciences. They will kind of get you to use skills from across the different papers, but they've given you the formula. So really it's just about putting the numbers in the formula. So we need to work out the distance. So that is 32 nanometers. We need it in meters because it wants our answer in meters per second. So we're going to need to convert that. That's going to be where the majority of these marks are going to come from. And then we've got our time, and that's 1.6 times 10 to the minus 7 seconds. That's already in seconds, that's fine. So all we need to do is convert the distance from nanometers to meters, and then we're going to divide one by the other, and that will give us our speed. So this is about knowing your kind of SI units and knowing your conversions. So we've got nanometers, then micrometers, then millimeters, then centimeters, then meters. I think going from micrometers to millimeters, we do a lot because of the magnification equation. So we should know that that's 1,000, the difference, or we divide it by 1,000 to go from there to there. In millimeters, there's 10 millimeters in a centimeter, and then there's 100 centimeters in a meter. And then for nanometers to micrometers, it's 1,000 again. So for each of these is divided by 1,000. So the easiest way I like to think about it, if you count up all of those zeros, if you just divide by 1,000, then divide by 1,000 again, then 10, then 100, there's 9 powers of 10 there. So it's 10 to the power 9. So we need to divide our 32 by 10 to the power 9 to get it to be in meters. So it's 3.2 times 10 to the power of minus 8. And that's how we're going to then have our number in the right unit that we need it for at the end. So then we just do a straight divide. So 3.2 times 10 to the minus 8 divided by 1.6 times 10 to the minus 7 gets us 0.2 meters per second. And now that sounds like a sensible number. Okay, so if you ever end up with a number that's really long, complicated number of decimal points and things like that in this, or if you times by 1,000, you'd get a really big number and things like that, then it's not probably going to be correct. But we've shown our working, so you could have done the jumpy thing that I did with that, write that out so that the examiner knows that that's how you got to your number as well. So as long as you put the numbers in the right way round, you get your division, you'll get your marks. If you used or you calculated that first 32 conversion wrong, but you used it in the right way and put distance divided by time, and you got a number, you can still get 1 mark for that. The government is trialling a new part of a driving test, which aims to help confirm if someone is still able to drive a car past a certain age. The trial method below was used to test people from age 18 to 65. First thing they had to do was open the program on the screen. When the screen changes colour from blue to red, the participant should click the mouse as soon as possible. The time it took them to click the mouse is shown on the screen. The program is reset to the blue screen. And then the participant will repeat those steps 3 more times. So they do 3 repeats, 3 clicks of the mouse, and then you would repeat all of those steps for each participant in the study. Give 2 reasons why using a computer program like this is likely to be more valid than the method involving dropping a ruler. So it's asking, why is this method better or more accurate than it would be if we were doing that ruler drop test? So you can think about some of the differences between them, or we can just try and learn some of these as a way of responding to this answer to one of these questions. But if you think about it, it is faster. Getting someone to sit at a computer and click a button like effectively 3 times, and then that's the whole test done, because it calculates the milliseconds of time for you with the reaction time. Then it's quicker than going through the whole ruler drop and converting it. You can measure it in milliseconds, so the computer can work out the reaction time in milliseconds, which a human can't do, so the resolution is smaller. So you can get an actual finer measurement on exactly or more closely to the real time it takes for them to react. And you don't need to do any calculations. So as we said, the computer does it for you. That means you're not going to look it up at a table. You're not having to read off where you think the thumb is on the ruler, get the best estimate for the centimetres or millimetres, and then go away and look up in a table. So that's open to error, human error from that calculation as well. It's also more random, so a computer is better at being random than a human. So if you're trying to randomly drop the ruler and not give any clues or hints, or try and not leave them waiting for the same amount of time between each test that you do, you're not going to be as successful at that as a computer is. So pick 2 of these and write them down. You've got some other kind of options in the mark scheme as well. When it talks about resolution, that's what it means about being able to do it in milliseconds rather than and being able to have a finer measurement. So it's faster and more random than relying on a person to drop the ruler. So there's my 2 points. The tests are designed to see if there are differences between people of different ages and to compare them. Give 1 variable the student should control so that a fair comparison can be made between the people of different ages. So by control variable, it means something that must be kept the same between the people that they are testing. Some people would make the mistake here of saying the age because you've forgotten that they're testing different ages. So if we're testing people of different ages, their age is changing. So that's not something we can keep the same because that's the independent variable. Dependent is their reaction time, that's what we're measuring, and we're talking about the control variables. So it's not going to be either of those. We have to think of something that has to be or should be the same between the people that they're testing other than their age that can then make the results comparable. So whenever we're thinking about control variables, we should always be thinking about what is it going to do that's going to affect the results. So this has to be something that could affect their reaction time if it was different between the different people. Sex could be one, whether they're male or female. Eyesight could be one, especially if you're having to get them to do anything on the screen or judge something with their eyes. So it could be whether if they need glasses, they should be wearing them. Not just whether they've got bad eyesight or not, but as long as they've all got glasses if they need them, so their eyesight is corrected to being perfect as well as it can be. Tiredness, we know that being tired can affect it, so that also there's an option in the mark scheme for time of day. So obviously later in the day, people might be more tired. Whether or not there's distractions, so if they can do it in like a soundproof room, or make sure they have the conditions around whether or not they're being distracted the same every single time. Whether or not they've had caffeine or alcohol, things like that we know can affect reaction time, whether it's increasing it or decreasing it. Any one of these will get you the mark. So that there's loads of options, but this is a good example of one of those questions where you have to think about control variables to do with people and to do with the things that could affect the results of the experiment, and then give some options. And these will always be, most of the time, on the list for options for control variables between people on the reaction time experiment, whether it's the ruler drop or this one. The table shows their results. So we've got the mean time in milliseconds, and then we've got the age in years. And it wants us to predict the mean reaction time for a 75-year-old. So with predict questions, often they may have a pattern in the table that you can use to help you. With maths, I know that when they say predict patterns in the table like this, there will be a regular pattern that you can then just copy. It's not always the same with science, so they want you to kind of use your judgment and the information from the table kind of together here to predict what you think is the most likely number. You're not going to be exact, so normally a range, or as long as it's above a certain number, as with this question. So if I go through and just have a look at the difference between these numbers, just to give me an idea, we've got 12, a difference of 12, a difference of 8, a difference of 8, another difference of 8, and then a difference of 7. So we've got this idea that it's got to be quite a big number. I would say it needs to be at least more than 8. And because the last one was 12, no, that first one was 14, and that last one was 12, I would say, especially as we know, we can see the pattern here, the pattern overall is that as age increases, the reaction time gets longer. So as this is another 10 years older person for 75, I'm expecting it's obviously going to be a larger number, and it should be at least 8, if not more, slower. 8 seconds, if not more, slower. So that's what I would say would be mine. You could, as long as you go, you could even go much bigger than 270 and you'll still be right. There's not going to be an upper limit. We just need to get the idea that, do we understand that it should be greater than 270? Yes. And then we've got to pick how much more than 270. And by going by this pattern, you could go, 8 more, 12 more, 14 more, and you get it right. Explain then, why is it more dangerous for older people to drive cars? Use information from the table to support your answer. So we need reasons why it might be more dangerous. So we're thinking about what's dangerous about a car. The danger is coming from potentially crashing or having an accident, either going on the curb or bumping into another car or bumping into a person. So this idea that we've got to link the idea that why is it more likely to have accidents, where there's more chance of having accidents potentially. Explain why, given the information you've got on the table. So we've got the pattern, which is as people get older, their reaction time gets longer. So it does say to use information from the table to support the answer. So what does the table actually show? It shows that as age increases, the reaction time increases. So how can we link this idea of slower reactions in older people to more accidents? Well, if they've got a longer reaction time, they might be too slow to brake or too slow to react to a change on the road, and therefore they're more likely to have more accidents. Because if you see something and you have to react to it quickly, so you have to put your foot on the brake because a person stepped out in front of you or a car is broken in front of you, then you're going to need to be able to have fast reaction times. So if they're not able to do that, then they're more likely to have accidents. So this is where we've got to combine for 3 marks what we can see in the table, the pattern, what that means about their reaction time, and link that to why they may have more accidents. So the table shows that as age increases, reaction time increases. This means that older people could be too slow or take too long to react to the changes on the road. This means they're likely to have more or cause more accidents. The thing that we need to make sure we've got in here is that we've got the pattern from the table, the idea that they're going to be too slow or they'll take too long to react, this idea that they're going to take too much time or the reaction will be too slow to be safe, and then the finally linked to accidents, which gets us talking about the danger aspect of why it's dangerous. Diabetes is a disease where a person is unable to regulate their blood glucose levels. Two people were having their blood glucose tested. One person has type 1 diabetes and the other is not diabetic. They drank the same concentration and volume of a glucose solution. Their blood glucose was then measured for the next 2 hours. Okay, so we've got a graph with the diabetic person, the non-diabetic person, and the changes in blood glucose concentration over 120 minutes. And it wants us to compare the changes in blood glucose concentration of the two people in the graph. So to compare, we need to compare the similarities and the differences. That means we need to give some similarities and give some differences. And it's asking about the changes in the blood glucose concentration. So how did it change over the time, the pattern over time, and what's different about those patterns, or what's the same? So my first big overall similarity is that both graphs rise and fall within the 2 hours. They both have their peak at the same time at 60 minutes. They both have the same delay at the start, so there's no change for 30 minutes and then it starts to increase at 30 minutes in both. So all of these are similarities. There's enough similarities there because if there's 4 marks, we need to kind of think about doing 2 of each. So now let's think about differences. So the diabetic person's graph is much higher. So the concentration is higher the whole time and the peak is also much higher. In the non-diabetic person, it returns to the start, returns to the normal levels before or by the time we get to 120, but that does not happen in the diabetic person. And then the other final obvious difference is you've got a really steep peak at the start and a much slower descent for the diabetic person, which explains why it doesn't return to normal in the same amount of time. So first I'm going to talk about my similarities. So in both people, the concentration rises and falls over the 2 hours, and the maximum blood glucose concentration for both was seen at 60 minutes after they drank the glucose solution. Then on to my differences. So however, in the person with the diabetes, their blood glucose concentration increased much more rapidly and decreased more slowly than the non-diabetic person. And then finally, I'm just going to add one more difference just to make sure that I've got everything covered because it's a nice easy one. After 2 hours, the blood glucose concentration of the diabetic patient had not returned to normal, but the non-diabetic patients had. So this was a nice one because obviously there was lots there that we could say, but just make sure with compare questions you've gone back and checked that you've talked about both similarities and differences. And remember, we were talking about changes. So we've used the word more or less or rapidly, more fast or slow. You can use the mark scheme to help you with this, but that's the kind of language we need. Using your knowledge of type 1 diabetes, explain why the graph looks different for the person with diabetes. So this is about using your knowledge. So that means you have to know and be able to explain what type 1 diabetes is. So type 1 diabetes is where they're not able to produce insulin to lower their blood glucose. So it wants reasons why to explain why the graph looks different. And obviously what is different is that the glucose levels are higher and they don't reduce as rapidly as in the non-diabetic person. So how can we explain that? What's not happening using our knowledge of type 1 diabetes? So the glucose is not being reduced as fast or as rapidly, and there is a bigger increase because there is no insulin being produced. So why is the glucose not being reduced as fast? Because it's not being absorbed by the liver because we don't have any insulin. So that's our 2 points. If you just said there's no insulin, that's not going to get you the full set of marks because we need to be able to explain that. We need to say that why is there no insulin? Okay, why does that mean the blood glucose levels doesn't decrease? Because it's not being absorbed by the liver, which is what insulin does. It causes the liver to absorb glucose from the blood. So my first point is because they cannot release insulin into the blood. That will get me 1 mark. So the glucose is not being absorbed into the liver cells. Long-term treatment for type 1 diabetes is a pancreas transplant. Long-term effects of type 1 diabetes, and we've got the long-term effects of an organ after an organ transplant. And it wants us to evaluate the option of a pancreas transplant for someone who suffers with type 1 diabetes. So this is an evaluate question. So we need to give pros and cons or advantages and disadvantages, and we then need to sum up with a judgment of our own. So we need to decide what we think is better, the pancreas transplant or not having it. So we're going to evaluate sort of pros and cons or risks in here. So we've obviously got a list of kind of negative effects or negatives or cons of having a transplant. And then all the things in this other column are long-term effects of having type 1 diabetes. So they're the negatives of having diabetes, which we could then flip around to say, turn them into positives of why you would need an organ transplant. What can the organ transplant do to decrease these? And this is the sort of thing that's often common with an evaluate question, is not just the information we're being given here, but we need to use our knowledge as well. So we're not just going to be able to basically take this information, just rewrite it out underneath as an answer to the question. It's going to be a leveled answer where we need to give our reasons for and against, link them together, make sure there's logical linking, and then also give our judgment at the end in order to get the top marks. So they all often do this, and they give you some information which gets you started, but you need to do something with this information or add your own knowledge together to get it to be a decent answer. So the long-term effects of type 1 diabetes, we can flip those around to positives of having a transplant, because you can live longer, you can have more energy levels because they'll have the insulin, so their glucose control will be better. They won't have to have any diet restrictions because they will no longer be injecting insulin. So you don't need to do that anymore if you have a pancreas transplant. All of these are then positive reasons for having the transplant. So the kidney failure and the loss of eyesight doesn't mean that having the transplant will reverse that or take that away. If they've already happened as a result of kind of the effects of long-term diabetes, then you're not going to be able to reverse that. But it shouldn't get any worse. It depends how serious those problems were before a transplant took place. So we have to think about the negatives of a transplant, so the opposite of that. Well, obviously we've got weak immune system and long-term medication here. We need to give more detail because we should hopefully know that the long-term medication is immune suppressants, and why we take them is because obviously there is a risk of rejection. So those are both two kind of negative, sort of a bit more detailed negatives of an organ transplant. And then we also need to think of something else, like if you're having a surgery, there's always the risk of infection. So we can always say that as a disadvantage for having a surgery. So then you put these together, and we have to try and think of ways we can start logically linking these to form a decent answer and then choose our judgment at the end. So I'm going to start off with my positives of the transplant. So the advantages of a pancreas transplant are that the patient would have increased life expectancy, along with a better quality of life, as they would have more energy and no longer need to inject insulin or have diet restrictions. So then I'm going to start saying what some of the negatives might be. So, however, there are risks with the transplant, including infection from surgery and risk of the body rejecting the organ. This can be reduced by taking immunosuppressants, but these must be taken for the rest of their life and can weaken their immune system, which puts them at risk of other infections. So this is where I've linked them together again. So I've said that there's a risk from surgery, there's a risk of rejection, but then I've linked that to the immunosuppressants that they have to take to help with the reducing of the rejection. But then I've also linked that to say that it's a negative because you've got to take it for the rest of your life, and that can be a negative because it can weaken your immune system. And then I've added the extra link there that means that you're at risk from other infections. So you can see how instead of just making that bullet points like it was in the table, so long-term medication, weak immune system, I've turned that into a statement that links those together. So why do you need to take the long-term medication? Why does that create a weak immune system? Why might that be a disadvantage for the person? The patient may also still need treatment for conditions caused by long-term diabetes before the transplant, for example, kidney problems or loss of eyesight. So I've kind of factored in the fact that they don't take those away. They might reduce the chance of them happening, but if they've already happened, then you're still going to need to have treatment for those. So they're my points. I've covered nearly everything we've said about a positive and a negative of both sides, and now I just have to do my judgment. Overall, I think that long-term, the transplant is a better option because although there are some risks, the long-term damage to the body will be reduced and the person could live longer and have a better quality of life. So then I've given my judgment, so I've chosen that the transplant is the best option rather than not having it, and then I've given my reasons. And I've said, although there are some risks, which I have said, the long-term damage to the body, so the kidney failure, the loss of eyesight and all the other things would be reduced because it would, and the person could live longer, which they could, and have a better quality of life, which sums up all of those things that we don't have to do anymore if you have a pancreas transplant that you would have had to do if you still had diabetes. So there's lots of different ways you can word your judgment, and it doesn't really matter what you choose. It doesn't matter if you said, I don't think the transplant's a good idea. I think, just treating diabetes with insulin is better. That's fine, as long as you've backed it up with reasons that make sense and are logical. The diagram shows an ocean food chain. So we've got phytoplankton, zooplankton, herring, and yellowfin tuna. And we've got their biomasses underneath. Identify the following from the food chain. A producer. So hopefully we know that a producer is a plant or a photosynthetic organism. It's always something that starts the food chain. So in this case, it's going to be the phytoplankton. Then it wants a predator. So again, definition of a predator is an animal that catches and eats other animals. So the only real options for that here are the herring, which can catch and eat the zooplankton, or the yellowfin tuna, which catch and eat the herring. So clearly we need to pick one of those two, so I'm going to go for the yellowfin tuna. So in case you're not sure, the yellowfin tuna is a fish, obviously, and then the herring is a smaller fish, and then the zooplankton are animal-based, tiny microscopic organisms that are in the plankton, and they eat the phytoplankton, which are the plant, tiny microscopic organisms in the sea. Okay, so some stocks of yellowfin tuna are being overexploited. Explain the impact of reduction in the number of yellowfin tuna on the zooplankton population. So this question is basically saying what will happen to the number of zooplankton if the numbers of tuna decreased, and why? So what would be the reason? So the herring, there would be more herring. So always start from the top and work your way down with these questions. So there would be more herring as there would be less tuna to eat them. So there'd be less of the herring being eaten, so there'd be more of them surviving, so their numbers would increase. That would mean that there would be less zooplankton because there'd be more herring to eat them. So more of them are being eaten because there's more herring. And therefore, our answer is what will happen to the zooplankton. Their numbers will decrease. Why? Because more herring are going to be eating them because less herring are being eaten by the tuna. So there's obviously a few ways you can write this. It's quite simply done in the mark scheme. So I'm going to write it as I would explain it. So the number of zooplankton would decrease because then we know we've definitely got that mark for that first point. There'll be more herring to eat them because there are fewer tuna eating the herring. And I've added that last bit because that makes sense to me and that explains how I understand what I'm talking about, but probably wouldn't necessarily need that last sentence to actually get the second mark. Human activity is impacting biodiversity. Peat bogs are unique wetland habitats for many species. Peat bogs are being drained for human use. Peat can also be cut into blocks and extracted as seen in the image below. Define a habitat. So this is one of the definitions we just have to know, but a habitat is the non-living part of an ecosystem. So it's the rocks, the soil, the ground, the water. Name one biotic factor that can affect a population in an ecosystem. So this time biotic means living, so it's a living factor or a living part of an ecosystem that can affect populations. So there's 4 to choose from. Now these are always the same 4, so it doesn't matter about whether it's asking you for one, multiple, it's asking you to explain or list them. The biotic factors that can affect populations, there's 4 that you need to know, 4 main ones that you need to know. So new pathogens, competition for whatever you would like to say competition for, food availability, and new predators. Or you can have hunting from humans, which is included in that, so you can't have that as well as predators. We only need one of these, so I'm going to go for new pathogens. But remember, I've got new in front of pathogens and I've got new in front of predators. The idea is that an ecosystem will have evolved along with a certain number of pathogens and the predators that are already there, and there'll be a balance to that ecosystem. If something changes, so a new pathogen arrives where organisms aren't resistant to it, or a new predator arrives which the organisms, the prey species, are not adapted to fight against or camouflage against or hide from or whatever, that can then drastically change the numbers of a population in an ecosystem. But there will be just pathogens that they're resistant to and they've evolved with and that don't affect the numbers greatly. And there'll be predators that they've evolved with that are in the food chain or the food web and that don't affect the numbers greatly, as long as the numbers are in balance. So just making sure we've got that word new in front of those if we are talking about a biotic factor that has a drastic change to the number of the population. Use your knowledge to explain why peat bogs have been destroyed by humans in the last 100 years and why it is important to prevent further peat bogs from being destroyed. So this is an explain question. It wants reasons. There's 6 marks available, so it's one of those long leveled answers. And we've got 2 bullet points to kind of guide us into 2 things or 2 areas that we have to talk about. And we're going to be able to use our own knowledge here as well as get clues from the bit of the question that was at the beginning. So we have looked at peat bogs and peat usage as part of land use in our content for the ecology topic. So you will hopefully know some stuff about peat to start with. But it says peat bogs are being drained for human use. So what are humans using the peat bogs for? Why are they draining them to get rid of the water? What are they using the land for? So it's going to be for things like farming, grazing of animals, building houses or roads and things like that. What are they extracting? What are they cutting it into blocks and extracting it for? Fuel, and as well as that, compost are 2 kind of main uses of peat, cut and extracted like that. So probably some of the easier things to say is, why is it important to prevent further peat bogs from being destroyed? So that's another way of saying, what's the impact on species there? Why is it important that we stop doing this and destroying this habitat? So you're going to get a mark for saying that it is habitat destruction because it says they're a unique habitat for many species. So by draining the water, we're making it not a wetland anymore. We're literally drying it out. So we're destroying the habitat. That's going to reduce biodiversity. That could then potentially affect food chains because you're reducing the number of species that can live in this habitat, which means you might be reducing the number of prey species or a predator species that can then affect the balance of a food chain. And also, a reduction in biodiversity is a bad thing because humans need biodiversity to provide them with resources. So we may think about the fact that there could be unique species there that could have potential as medicines, or we don't want to make sure we don't lose or cause any species to go extinct because they could be useful to us. And then another reason why it's important to prevent this extraction and destruction is because the extraction actually contributes to global warming. So it releases carbon dioxide into the atmosphere, contributes to global warming, which contributes to climate change, and that can have another impact on biodiversity as well. It also says why they've been destroyed by humans in the last 100 years. So yes, this is what they've been destroyed for: farming, grazing, building, fuel, compost. But why? And that's because it's about more people. So remember, when we looked at land use and pollution, all of those things, we talked about kind of why humans are having a greater impact, and it's because the population is increasing, and therefore we need more land and more space to grow crops to feed more people. So that's the reason why they've been destroyed, as well as how they've been destroyed. So reasons why peat's been extracted for fuel and for compost, reasons why it's been drained: farming, grazing, building houses, whichever you want to use, and then the kind of reasons why those have been getting worse, or why more peatland's been used for that, is because of the increase in population. So more people means more land, or more need for fuel, or more need for food. And then the reasons why to stop, we're going to talk about the fact that it's a destruction of habitat, we're going to talk about the impact on biodiversity, and we're going to mention the impact on global warming. So that's my kind of mini plan. This is exactly what I would do if I was in the exam before I wrote it, because it's a lot to write. So here's my answer, and as I read through, I'll highlight where I've kind of put the bits I said I would in my plan. So in the last 100 years, the human population has rapidly increased, and so more land has been used for farming and housing. Peat bogs have been drained so that there is more land to grow more crops and graze more animals to feed the growing population. So this is kind of why it was drained and why the land was used, and I've mentioned the human population. Peat has also been extracted for use as a fuel and as a compost to grow plants. So that is why it's being kind of cut out and extracted. Further destruction of peatlands must be stopped, as it is destroying unique habitat for many species, so it could lead to their extinction. This will reduce biodiversity in the peat bogs, which can disrupt food chains and the ecosystem stability. The extraction of peat also releases carbon dioxide, which contributes to global warming, which can cause a reduction in biodiversity. It is important to maintain biodiversity as humans rely on species for important resources, such as pollination or textiles. You could have talked about medicines or the potential for discovering new medicines in the future or something like that for the last part. But you can see how I've definitely covered both of those bullet points. I've linked ideas together. So this idea of the usage of the peatlands, so for more food and more kind of growing crops or grazing animals and stuff like that. But I've then linked that to the growing population. So why in the last 100 years specifically, it's got worse, and it's because our human population has rapidly increased. Then I've linked to this idea of how further destruction of peatlands could keep or continue to reduce biodiversity, and then explain why that's a bad thing. So the destruction of food chains, the releasing of carbon dioxide and the global warming, which even more continues to reduce biodiversity, and then the reason why biodiversity is important, other than just we don't want to cause species to go extinct, but because that can then cause us to not have those important services.
Well done for making it to the end of this paper, lovelies. I'm really, really proud of all of your hard work.